University of Hawaii. And then we may or may not be like introducing speakers in between there just based on the interest of time. So here we go.
So I'd like to say thanks for inviting me. I think this is a really timely Spotlight session to be doing. I'm excited to see the rest of the talks. Today I'm going to be talking about some work that I've been doing in collaboration with Lyndon Coghill and Jeremy Brown, both at LSU. And this work really focuses on trying to tease apart sources of conflict in big phylogenomic data sets. So if you saw Anna Yoder's presidential talk yesterday, she made this point that maybe as a field we sort of got into phylogenomics with this expectation that it was sort of naive. The idea was maybe we can take hundreds or thousands of loci and glue them all together, and that will somehow average over the conflict or sources of error in our data sets, allowing us to achieve consensus and overcome uncertainty in phylogenetics. We now know this is not true. We're all familiar with the ctenophore problem, whether ctenophores are sponges or sisters of the remaining metazoa. This is a case where we have multiple replicated genome-scale data sets that say fundamentally different things about the tree of life with maximal support. This is not uncommon. We can all think of examples from our own areas of work. What I'm going to focus on today has to do with the phylogenetic position of turtles. So what I'm showing you here is the backbone phylogeny of amniotes. This includes all the major lineages except turtles. And so this phylogeny has virtually no uncertainty in it at this point. No one really argues about this tree anymore, except where turtles go. So over several decades now, people have tried to answer this question, where turtles go in this tree, using a variety of sources of data, from morphology to mitochondrial DNA to nuclear DNA, using lots of different creative types of analyses. So the dots on this tree indicate that at least one paper has been published using reasonable data, reasonable methods, that come to the conclusion that turtles go there. Right? So we have a lot of uncertainty about this. Given this uncertainty, it's not at all surprising that people have tried to target this question using phylogenomic data sets. So at least 3 placements have been published using really large format sequences for turtles. I would say 2 of these placements are still totally credible. So we still really don't know. And this is what got me and Jeremy and Lyndon interested. We started looking at these data sets as sort of a test bed to try out our phylogenomic methods. So all of this interest has led to replicated data sets being assembled for this same question. And these range from hundreds of markers assembled from transcriptome sorts of data, around 1,000 ultra-conserved element loci, just dozens and dozens of Sanger loci, on up to orthologs mined out of whole genome sequences at this point. So we've started using this as really just a nice model system for trying out different ways of doing phylogenomic analysis and really trying to assess the performance of methods. So today I'll be presenting just a small fraction of that work we have going. Okay, so the first thing we did, just the most basic thing you can do with this sort of data is say, okay, let's take the biggest data set in terms of number of genes, and then maybe the biggest data set in terms of number of taxa, and analyze it gene by gene. Do a nice analysis of those gene trees and ask, do we see concordance? If this expectation that we had going into phylogenomics holds, we might expect that most of those gene trees should agree with one another, and maybe there will be some variance around a most common gene tree that's owing to low power or minor issues here. That's not what we see. So these pie charts correspond to the number of genes, or the proportion of genes that recover different placements for turtles for 2 different data sets. This one comes from genome scale data, and this one comes from Sanger sequencing. The colors correspond to placements on the tree next to it. So we see in both cases, the most common placement for turtles is either sister to archosaurs or sister to crocodilians. but there's a lot of variation. Right? We're not really even close to consensus. Most troubling from this data is the white part of the pie. So these white trees, the other, are the gene trees that don't recover the amniotic backbone, that virtually everyone agrees is one tree. Right? So we see a lot of variation, more than we went into this expecting. Something must be driving this. So today I'm going to present some of our work trying to dig into this a little further. So I'm really just presenting the results of two fairly simple analyses. The first I've already told you about. We just went gene by gene and did a nice Bayesian estimate of the gene tree. And what that gives us is a posterior probability for every node in the tree. The second thing we did was calculate a marginal likelihood for the presence of every node in the tree, and then a marginal likelihood for the absence of that node in the tree. And by taking the ratio of those two things, we can now calculate a Bayes factor for each node in the tree. So we do this for every locus and every data set, and it lets us start doing things that at least we found pretty useful. So right off the bat, we start noticing things like this. When we're focusing on posterior probabilities, as we tend to do in phylogenetics, we're naturally bounded. These things range from 0 to 1, and we all know that it's super common to see these values achieve either their maximal or minimal value. Right? So at least for this relationship in this data set, 26% of the genes have either a posterior of 1 or 0. Okay? So that's fine, but it obscures a lot of information. So these two genes have identical estimated posterior probabilities, and yet their Bayes factors are very, very different. Another way of saying that is we have vastly different odds of observing those sequences if archosaurs and turtles are monophyletic. And so by focusing on this bounded measure of support, we're actually missing a lot of the important differences in information. Okay, so the second big message that came out of this is that a very small number of genes can drive our results. And so one of the big promises of phylogenomics is by gluing together hundreds or thousands of genes, we're lessening the impact of any single gene and hopefully averting problems. So what I'm showing you here is a violin plot with distribution of Bayes factors across genes for each node. Okay, so to break this down a little further, on the right side of the plot, we have the backbone nodes for the amnio tree. And we can see things that sort of make intuitive sense. So things like monophyly or crocodilians. All of the genes have sort of moderate to high positive Bayes factors. So there's a lot of information in the data that says crocodilians are monophyletic. That sounds about. And that's basically the case for most of the backbone relationships. A few are more uncertain, but overall, we find positive support. On the other side of the chart, we see something different. So these are all potential places we can stick turtles into this tree. Now, if there was strong information in the data, one of these should be predominantly above the line, and the rest of them should all be below the line. And that's not really what we see. What we see is most of them are centered right on the line. What that means is the majority of genes in these data sets are effectively silent on where turtles go in the tree. If we have to pick some that look best, if we want to make a guess at this point about where turtles go, it looks like the most positive ones are turtles are sister to archosaurs, perhaps turtles sister to crocodilians, or maybe sister to birders, but this is pretty nuanced, teasing things apart. There's no clear signal. Okay, so if we take this data set, this is the EST data set made up of 248 loci, published by Chiari et al. If we just concatenate those and get a tree, this is what we get. Turtles are sister to crocodilians with a posterior probability. So you do that, and then of course you zip back to our distribution of support across genes, and we notice that the crop result actually looks a little different than the others in an important respect. It has this spike up top. What that spike turns out to be is two genes in this data set of 248 that very, very, very strongly say turtles are sister to crocodilians. The rest of the genes in the data set don't say that, or they say it very weakly. So a natural thing to do at that point is to go from 248 loci to 246 loci and redo the analysis. If you do that, turtles jump over to being sister archosaurs, again with posterior probability. Okay, so we're saying fundamentally different things about when this major lineages of amnio arose. This has implications for how the shell evolved, how all sorts of morphological features evolve. So that's a troubling result. Unfortunately, it seems like it's not uncommon. Okay, so we have since found a similar pattern in one of these other data sets. One of the data sets derived from full genome sequences, where we can remove less than 1% of the genes of a data set of 1,100, and we get the same result. Moreover, 2 more recent papers have come out that build on this work and follow up on it, looking at other sorts of data. A paper by Walker et al. Is up on bioRxiv now that finds more outliers, and a paper by Shen et al. That came out in Nature Ecology and Evolution maybe a month or 2 ago looks at a larger number of data sets and finds qualitatively similar results. Okay, so the next question is what's causing this? Why do we have these outliers? At present, they don't seem to be particularly uncommon. What are they? So to start digging into this, we just went to those genes and started playing around. So one of the things we did is pull all of the sequences out of those alignments and blasted them against their own genomes and just saved all of the significant blast hits. And then you can reconstruct the phylogeny with sort of extra gene copies in there. What we see is pretty clear evidence that at least these 2 genes we found are parallels. So here's that tree. The bold tips in the tree are the sequences in the initial alignment. The other tips in the tree are significant hits to the genomes of these organisms. And we can see pretty clearly that there's 3 copies of this gene, 3 parallels. And if you look at what those taxa actually are, it turns out that we're sampling 1 copy of this gene for crocodilians and turtles, and another copy of the gene for all the other animals. So the true history for this gene really is, or the true history for this group of paralogs really is that turtles are more closely related to crocodilians. But we've just missed that in assembling our data set. It's just an error in the data set assembly. The other gene turns out to be the exact same thing. We find 2 copies here. We happen to sample crocodilians and turtles for 1 gene copy and the other amniotes for another gene copy. So we're continuing to dig into this. This is the first 2 outliers we found. So far, the source of these seems to be paralogs in our work. Now, I wouldn't claim that that's going to be common. I think paralogs are likely just really easy to detect. There could be other things driving this. In that Walker et al. Paper, they found 1 outlier gene that had no clear evidence of chirology, had no clear evidence of other sorts of prosaic error, and they suggested that this gene may be a genuine but rare product of the evolutionary process. It's still sort of an open question. Shen et al. Has found the most outliers to date, but they didn't go into causes, at least not yet. So really, this is still an open question. Are these mostly analytical problems, issues with how we assemble our data sets, or are these biological problems, driven by incomplete lineage sorting or something like that? This really needs work. So, to wrap that up, I would suggest that this is a major priority for the field going forward. A result like this, these pie charts, these aren't all parallels, right? This is a complex mixture of different problems that lead to a pattern like this. So we now have straightforward methods to detect these problematic genes. You can do this with Bayes factors like we did. You can do this with likelihoods. So you can do this, and if you're working with big phylogenomic data sets, I would really suggest that you do. But what we really need are better, faster methods for identifying the actual sources of these problems that will allow us to screen them out in the first place and identify them. Okay, so I'll stop there. I'd like to thank David and Emily for the invitation and then funding and computation services. Thank you. And it looks like I have about a minute for a question. Yeah, Adam.
Thanks, that's really interesting work. I have sort of a 2-part question. Does it matter what kind of estimator you use for the marginal likelihoods? And then if it does, do you have any recommendations for us on which one to use?
So we did this all using stepping stone integration. We didn't compare it to any others. I would think doing stepping stone integration and using that would be much better than maybe the harmonic mean or something. But we didn't look at different ways of doing it. Did you have a second part to your question?
I thought that was 2. but it's really early.
Okay, thank you.